thank you very much for getting up so early to hear about Building Immune Defence Priority Research Program. This is what I like to call the Trinity of Health. An appropriately functioning immune system is critical for your health and well-being. Conversely, an impaired immune response results in impairment of both your physical and your mental wellness and leads to reduced productivity. As I mentioned in the video, nutrition, next door to vaccination, is one of the leading ways we can impact the immune system. The leading cause of immunodeficiency, or when the immune system doesn't work appropriately, is malnutrition. Conversely, the leading cause of inflammation, as we heard yesterday, is overnutrition or obesity. And today I'm putting forward our hypothesis that one of the vital connections between nutrition and the immune system is the minions, as we heard yesterday, the 100 trillion gut microbiota that live predominantly in our gastrointestinal tract, alongside 70% of our immune cells that at any one time we can find in that gastrointestinal tract. We can directly impact those immune cells through food, and they will use the immune cell highway, otherwise known as the circulation and the lymph system, to traffic anywhere else in the body. Our target Asian export market are consumers who have a high cultural index. These are people who believe in the value of a hard day's work to improve themselves in life. And they also seek food and beverage solutions to maintain their health. We have two research projects as part of Building Immune Defence, and that is looking at building immune defence and food health relationships for respiratory tract infection, and also food health relationships for respiratory defence against pollution-driven inflammation. The understanding of immunity from a consumer awareness point of view is at an all-time high. Surveyed 77% of Chinese consumers rated immune enhancement as important, and this is because of the recent outbreaks of SARS, the bird and swine flu, and the ageing Asian population. It's also because Asia's air pollution crisis is presenting a significant threat to human health. And we believe that these perspectives suggest a high potential adoption rate of food and beverage solutions targeting respiratory immune defence. To achieve our research objectives, we have a large team. We've pulled together world-leading researchers from all over New Zealand to deliver our research outcomes. The immunology for the Building Immune Defence against Respiratory Tract Infection will be delivered by my colleagues at the Mulligan Institute, and our clinical collaborators at the Medical Research Institute of New Zealand will deliver our exploratory study. Wayne Young at Ag Research is our bioinformatician extraordinaire who's going to bring all this amazing big data together to allow us to identify the differences in immune responses in our cohort. Sally Poppet and her team at the University of Auckland will deliver the detail from the food diaries that we will gather as part of our exploratory study, and Amber and Sarah are our nutritionists, or our dietitians I should say, on the ground working with us as part of the exploratory study. And of course we have our colleagues at Plant and Food Research who are leading the Building Immune Defence Against Pollution-Driven Inflammation project as part of this program. And this is all possible because we have the pillars of consumer insights and the science of food bringing this together so that we can find these food solutions. The overall goal of Building Immune Defence is to fast-track opportunities for New Zealand companies to develop foods and beverages with scientifically validated immune defence benefits. But we have some gaps in our knowledge, and first we must address the causal relationship for the beneficial modulation of immune defence. We need to identify nutrition responsive biomarkers, both established and new, that will tell us both physiological and immunological processes. And we will examine the efficacy of these biomarkers to respond to food interventions in our mouse models. So my colleagues at Plant and Food will outline Building Immune Defence Against Pollution-Driven Inflammation in the next talk. I will focus on building immune defence against respiratory tract infection now. As we mentioned, the Asian consumer are often wearing masks. Outside for that pollution, as will be introduced in the next talk. Inside, what are they afraid of? They're afraid of this. And that's because influenza epidemics are occurring almost every year. And the link between the physiological and the psychological impacts of getting a cold or a flu infection are clear. We all know the physiological responses where you're sneezing, you've got a runny nose, you've got a fever, but in turn we also understand that lethargy in the brain and not being able to think properly, that cognitive impairment that occurs when you have a flu infection. And we're talking about consumers who are demanding solutions to treat this so that they can improve their productivity and they can get to work for a hard day's work. And as a consequence, if we use China as an example in this market, 75% of Chinese individuals are prescribed antibiotics for a flu infection despite the lack of efficacy. And if we put this into an actual amount, what this means is the Chinese consumers are having, on average, 10 times more antibiotics than an individual in the US. 138 grams per person per year in antibiotics consumed. And we heard from Stefan yesterday about the problem of antibiotic resistance, but there's a just as great an issue because antibiotic use screws up your immune system. So there we've got accumulating evidence that suggests that antibiotic use will perturb immunity against influenza. So we're not only taking something that has no efficacy, it actually likely has a negative outcome. How do we know this? Well, this has come from studies in mouse models. So here we are looking at animals that have been infected with influenza A virus and treated with antibiotics. Two studies. In the left-hand side, we have protective antibody titer, and you can see both IgG total and IgG2A is significantly impacted in animals that are treated with antibiotics. On the right-hand side, again, influenza A infection, antibiotic treatment. We've swapped the colours around from black and white, but you can clearly see that that protective antibody response, which will stop you from getting an influenza infection again, is markedly reduced in these animals that are treated with antibiotics. And the same is true for vaccination. So on the left-hand side, in the black, we have animals that have been vaccinated with the seasonal flu vaccine, just like we all get every year, and in red, it's mice that have been treated with antibiotics. And over time, we're looking at the development of the antibody response in these animals, and you can see that it's been significantly impacted. The protective immune antibody response driven by the flu vaccine is perturbed in animals treated with antibiotics. And on the right-hand side, we're looking at that protective antibody titer again at day 28 when we see the peak in that response. In mice that lack the ability to see flagellin, which is what the motile bacteria use to move around, there is a significant reduction in their production of antibodies. And importantly, if we take germ-free mice, recolonise them with microbes, we can see that their immune response is back to like a normal mouse, suggesting that the gut microbiota play a critical role in protective immunity to influenza. And in a somewhat ironic twist of fate, we were always trying to make mouse into man. Here, this is a study looking at almost making man into mouse. So it's repeating those studies in humans, taking individuals, giving them antibiotics and the seasonal flu vaccine, and examining how this impacts the response in a human population. We intend to take a different approach because we're not looking at targeting a consumer market of antibiotic users when you get the flu vaccine. We're looking at targeting the normal population. So we understand that these are individuals who are demanding solutions to improve their immune response to an influenza infection. What we want to do is change that antibiotic panacea to food and beverage solutions that can improve their immune defence against influenza. And for those of you in the room somewhat hyperventilating at all this gut microbiota talk because you think I'm drinking potentially a little bit too much of this Kool-Aid, fear not. Whilst we have a strong hypothesis that the gut microbiota may play a role in building immune defence, I am very well aware that we have a large number of foodstuffs and their metabolites that are known to impact the immune system. So we will be looking for foods around gut microbiota, but that is not the only source of food that we will be looking to beneficially modulate immune function. We've chosen vaccination because, in essence, it provides a proxy of a wide variety of immune responses. So if we're to boil vaccination down to its simplistic advantage for us, it's a standardised immune challenge that we can measure the response to. It's also increased numbers of individuals attaining acceptable antibody titers is an acceptable nutrition responsive biomarker. Sounds quite easy, really, when we put it like that, doesn't it? However, we still don't know how we're going to beneficially modulate immune function, and that's the gap in knowledge that we need to address with this research. The first tranche over the next 3 or so years of research that we are going to deliver, we have these 2 underpinning pillars that will allow us to achieve our goals. The first is a very diverse and interactive industry reference group who we will work with to decide food and beverage candidates that we will identify across the program to test to see if these can build immune defence. We will also work in collaboration with our consumer insights and science of food colleagues so that we can develop, design and test potential food products. As I said, this research program is split into 2 very interesting research projects. For terminology, we call them Objective 1 and Objective 2. Objective 1 is building immune defence against respiratory tract infection. The first thing we need to deliver is an exploratory study looking at human participants and their immune response to the seasonal flu vaccine. We'll be examining the association of gut microbiota and that immune response as a part of this objective. We will also be taking that systems immunology approach that I briefly touched on earlier, with Wayne Young being able to pull together all of this information: gut microbiota, immunology, antibody, food diaries, all of the information coming together to identify how people respond differently in the normal population to the influenza vaccine. We'll also have preclinical mouse models of impacting the gut microbiota and examining how this affects immune defence to the seasonal flu vaccine, and we will be examining lead food candidates within those models to see if we can establish food solutions that beneficially modulate immune function. Objective 2, as mentioned, led out of plant and food research and will be discussed in the next presentation, is developing preclinical models of pollution-driven inflammation and examining how we can modulate the immune function and using, again, the lead food candidates identified to beneficially modulate respiratory immune defence against pollution-driven inflammation. One of the biggest challenges we face is that the immune response varies from person to person. So if we were to give you all seasonal flu vaccine today and measure your immune response, there would be marked variability in the protective antibody that is made in all of you healthy, normal population. Data to date suggests that non-heritable factors have the greatest contribution to immune health and well-being. We know that immune response to vaccination depends on baseline immune status, so what you look like today will present how you will respond to that immune vaccine. It allows us to identify people in the normal population who are perhaps low responders to be able to do our food studies in. And importantly, baseline immune status is stable over an extended sampling period. So the bottom publication that's just come out in the last few weeks suggests that only 1.4% of variation in individuals over their homeostatic or their immune steady state. So what we need to focus on is the variation between person and person. Can we identify the low responders who will be able to take beneficial foods to improve their immune response, and how do they compare to a high responder in the first instance? So we've already started our exploratory study. This advert made the cutting room floor. It didn't go out to the public, but it's one of my favourite ones, to look at the association of gut microbiota in this response to the seasonal flu vaccine. And this is what we're doing. We're looking at 125 participants aged 18 to 64 years. We're getting fecal samples, food diaries. We're also employing the American Gut Lifestyle Questionnaire. These individuals will be vaccinated. We will take several blood samples across time to examine their immune response to the seasonal flu vaccine. So we'll have a fecal sample prior to immunisation that will go along with a food diary. We will take a sample of blood prior to vaccination. That's our baseline response that we can measure everything back per individual. We will also be taking blood on day 3 and day 7 to be able to capture exactly what the immune response is doing straight after vaccination. It should provide us with new and early predictive biomarkers. Day 28 is when we're going to measure those protective antibody responses. We'll also have another fecal sample to go along with that and a food diary. And we'll also be taking samples at 6 months to examine the persistence of that protective antibody response. As I said, we've already started the exploratory study. We rolled out a test group on the 1st of April. Everything went well. So this week we began the full rollout of participants. It's my understanding as of yesterday we had put through approximately 39 participants through their day 0 time point. And this is one of the largest studies of its kind, incorporating all of this immunological information. And that's because the team will process in under 3 months 26 litres of blood into immune cells, and we will have collected 250 samples. And just so you can see all of the parameters that we'll be measuring across the days, the HAI, or hemagglutination inhibition assay, is a functional readout of those protective antibody responses. So we'll have a baseline, we'll have the crucial day 28 point, and at 6 months, so that we can measure the persistence of that response. We'll have the microbiota and food diaries associated with baseline and with that antibody response. We'll be looking at gene expression in those early few time points to identify new biomarkers of the immune response, which we will be able to use in our future food trials, and we'll know within a week whether or not we're seeing efficacy. We'll also be doing a bunch of other immunological tests, looking at flow, antibody production, and specifically at the immune cells that make the antibody, the B cells. The T cells are the ones that tell them what to do, and putting all of this information together in that big data way, the systems immunology, to see if we can identify low responders who could do with improving their immune response to use in a future trial. So the outcomes that we're looking for is we're looking to assess intra-individual responses as compared to baseline. So that variation across the cohort, what does it look like? Can we identify low versus high responders? We'll be looking at inter-individual diversity, Stool community types within this New Zealand cohort for the first time, and again, for the first time worldwide, the association with gut microbiota community types and influenza vaccine responses. So we're really looking for that high-low responder profile. And on the right-hand side, the graph you can see here, 31 individuals, their immune response to the flu vaccine. That response ranges in a difference from 1-fold to 128-fold from their difference of day 0 compared to that day 28. What is it that's making them so variable? We propose it could be the gut microbiota. And we'll also be identifying biomarkers that can allow us to predict this immune response. Here we're looking at the Stool community types. Previous investigators have identified enterotypes that we can break individuals down to 4 specific enterotypes based on the gut microbiota that they have. Does one of these enterotypes allow us to predict high antibody responders versus another one that may associate with low antibody responders? And this is the systems immunology approach that we will take. So we'll be looking at cell populations. Here's the measure of the B cells that make the antibodies. Gene expression. How do we identify these responders and identify those nutrition responsive biomarkers? The antibody response will be taken into account. Are they a high responder? Are they a low responder? The baseline signatures, what tells us about that person? If the baseline response is what predicts how they respond to the flu vaccine, who are those people in those baseline responses? How do we identify them? What's the specific immune footprint, so to speak, that will allow us to identify those individuals in the normal healthy population? And of course, the Stool community type. Our overall goal is to examine whether there's an association to Stool community type and protective immunity to influenza via the seasonal flu vaccine. We're also working on animal models to understand how the gut microbiota may impact immunity to flu. On the left-hand side, what we have is 2 sets of mice that are genetically identical, but they have different gut microbiota. The blue mice are one set, the red are another, and you can clearly see if we give them the seasonal influenza vaccine, there's a marked difference in their ability to respond, and the only thing that's different about these mice is their gut microbiota. We've also replicated the studies that have been done overseas, given mice antibiotics and shown this negatively impacts their immune response to the seasonal flu vaccine. And importantly, we can tailor that antibiotic delivery to impact the immune response. Therefore, we can use these models to see which one is most like our human data and use this to get mechanistic understanding to work out how we're going to beneficially modulate immune function to the seasonal flu vaccine. We also have what we call the 21st century dysbiosis. We've heard a lot over the last day about how we can impact our health through poor diet, through stress, and through early antibiotic use. Can we generate an animal model that does all of this, just like what happens in the 21st century, and examine how this impacts the immune response to influenza? But importantly, whether it's this model or the previous two that I showed you that are most associate with what we're seeing in our human cohort, the overall goal is the nutritional intervention to assess these food candidates from New Zealand industry partners to see if we can beneficially modulate immune function and measure those immune responses and provide that causal relationship for a food health claim. So this just leaves me to thank all of the people that have been involved in getting this research program together. Importantly, the HVN management team, and I'd like to thank John, David and Ephlam in particular for allowing this project to get up and running, and of course Eleanor and Ely for bringing us all together in this spectacular way for the program over the last couple of days. The science leadership team and the gut and immune expert panel, scientists from all over the country came together to bring together ideas and deliver what we thought was the best way forward. Consumer insights and science of food, they're allowing us to do what we intend to do to be able to deliver these food products. And of course the Immune Defence Industry Reference Group. As I mentioned earlier, we have a diverse and very engaged group of industry, so thank you very much for your time.